Hi everyone, today we are going to learn about glycolysis and the entire pathway, its energetics and regulation. Everything will be dealt within this single page. So let's get started. First of all, what is glycolysis? It is the metabolic pathway where glucose is broken down into pyruvate. And what is its site? It occurs in the cytoplasm of the cells. There is no oxygen requirement in glycolysis. Okay, so when there is unavailability of oxygen, that is an anaerobic condition, the energy that the cells get is through this metabolic pathway only. Now let's see the entire pathway in details. First of all, glucose, it is converted into glucose 6-phosphate using the enzyme glucokinase or hexokinase. Now when glucokinase takes play or comes into action and when hexokinase comes into action, glucokinase is present in the liver cells and hexokinase is present in brain, in the cardiac tissue, in the skeletal muscles. Now why is it so? Glucokinase, it has a high Km value, that is a high Michaelis-Menten constant. What does high Km value indicates? It indicates that more substrate is needed to reach the Vmax by 2, that is the half of the velocity of the reaction. Remember the graph of enzyme kinetics? So more is the Km value, less is the affinity of the enzyme towards glucose. So it will act when there is adequate supply of glucose is present, so that excess glucose can be stored. Where adequate glucose supply is present in liver, so glucokinase is going to act in liver. And hexokinase, it has a low Km value, so it has more affinity for glucose. So it will act when there is less concentration of glucose present, so that it will be very efficient in taking up the glucose and pushing it down into the glycolytic pathway. So hexokinase is present in brain, so brain tissue needs glucose, right? If there is not sufficient glucose, it can reach to coma state. So brain, cardiac muscles, skeletal muscles, here in majority of the cases hexokinase is present. So now let's continue. So glucose to glucose 6-phosphate, one phosphate group is being added, so a ATP is broken down into ADP. Now, so this process, it locks the glucose in the cell. Okay, and after that, from glucose 6-phosphate to fructose 6-phosphate. Now the first, very important, in this entire glycolysis, there are the 3 steps which is irreversible. The first step, the third step and the last step. So these are the 3 steps which are irreversible in glycolysis. You need to keep this in mind. So this first step, it is irreversible and it uses 1 ATP. Next, coming to then from glucose 6-phosphate to fructose 6-phosphate, there is no change in the molecular formula, so it will be an isomerase enzyme, right? So it is a phosphoglucoisomerase. You can get the name of the enzyme from its action only. Just be very careful and you will get it. Next, coming to fructose 6-phosphate to fructose 1,6-bisphosphate. Again, one phosphate group is being added, so 1 ATP is used, and this is done by the enzyme phosphofructokinase. Phosphofructokinase, it is also called the pacemaker enzyme. This is the rate determining step of the entire glycolytic pathway. That is, the velocity of this pathway will determine the rate of the glycolytic pathway. Hence it is called the rate determining step. Just like in chemistry you have learned about the limiting reagent, similar thing is this, the rate determining step. So it is also called pacemaker enzyme, and this is the phosphofructokinase. Then from the fructose 1,6-bisphosphate, it is converted into by aldolase enzyme into dihydroxyacetone phosphate and the glyceraldehyde 3-phosphate. right? It is also an isomerase reaction, the interconvertible reaction between dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. It is done by triose phosphate isomerase. Now majority of the compounds, it is present in glyceraldehyde 3-phosphate form. Then from glyceraldehyde 3-phosphate there is 1,3-bisphosphoglycerate. So there is again, what is happening? See, there is glyceraldehyde 3-phosphate and it is becoming 1,3-bisphosphoglycerate. That means the hydrogen is getting removed and a reducing equivalent comes into play here. So this is a dehydrogenase. So it is a glyceraldehyde 3-phosphate dehydrogenase and a reducing equivalent of NADH is produced. This NADH can go into the ETC, that is the electron transport chain, and can produce ATP. We will see this in the energetics part. Then 1,3-bisphosphoglycerate, it converts into 3-phosphoglycerate. Now 1,3-bisphospho. There were 2 phosphate groups. Now there is only 1 phosphate group. So ATP is getting produced in this step. So this is the energy generating step. The first step where hexokinase and glucokinase were used, that was the energy utilizing step. Again, in the step of phosphofructokinase, that was also an energy utilizing phase. But in this step, that is from 1,3-bisphosphoglycerate to 3-phosphoglycerate, this is energy generating step. Now, from 3-phosphoglycerate, there is 2-phosphoglycerate. Again, there is no change in the molecular formula. It is a mutase. Only the position of the phosphate group is changed here. That is from 3-phosphoglycerate to 2-phosphoglycerate. So it is a phosphoglycerolomutase, and from 2-phosphoglycerate to the phosphoenol pyruvate using the enolase enzyme. From phosphoenol pyruvate to pyruvate. This is also an irreversible reaction of the glycolysis. This is the third irreversible reaction of glycolysis. It is done using the enzyme pyruvate kinase. Okay, and this is also an energy generating phase. Now see, phosphoenol pyruvate, there was a phosphate group here, but it is converting into pyruvate. So phosphate group is being released in the form of ATP, right? Now this pyruvate, if this pyruvate enters into the link reaction and then the TCA cycle, then there will be an oxidative if it enters the oxidative pathway, then using the TCA cycle it can enter the citric acid cycle. Otherwise it can also be converted to lactate using the lactate dehydrogenase. These are the various fates of pyruvate. Now let us see the energetics first. So suppose it is in the aerobic condition. So in aerobic condition, hexokinase or glucokinase, there is an energy utilizing phase. So number of ATP gained will be -1 because ATP is getting utilized. So we are writing here -1. Phosphofructokinase, again ATP is utilized. So number of ATP gained -1. Glyceraldehyde 3-phosphate dehydrogenase. Here we saw that yes, NADH is getting produced. But remember, this glyceraldehyde 3-phosphate is a 3-carbon compound and it is coming from fructose 1,6-bisphosphate. So there will be 2 glyceraldehyde 3-phosphate. So for 2 glyceraldehyde 3-phosphate converting into 2,1,3-bisphosphoglycerate, there will be 2 NADH generated. And we know that from 1 NADH we get 2.5 ATP if it enters into the electron transport chain. So from 2 NADH we are going to get 5 ATP. Then coming to 1,3-bisphosphoglycerate kinase. In kinase, again there are 2 molecules, 1 ATP. So this ATP is also very important. That is these 2 steps where ATP is directly generated. This is called the substrate level phosphorylation. So substrate level phosphorylation occurs between 1,3-bisphosphoglycerate to 3-phosphoglycerate and phosphoenol pyruvate to pyruvate. So here 2 ATP. Also in case of pyruvate kinase, 2 ATPs are produced. So total it is 9 minus 2, 7 ATP. So 7 ATP is generated through this glycolytic pathway in aerobic condition. But if there is anaerobic condition, ETC will not work. that means that ATP that we are gaining from the NADH part, we will not get that. So in that case there will be 4 minus 2, that is 2 ATP. So net gain will be 2 ATP in case of anaerobic condition. Now coming to the regulation part. In the regulation part you can see that yes, it is very clear and it is self-explanatory. Insulin is stimulating glucokinase, glucagon is inhibiting. The products, that is glucose 6-phosphate, fructose 6-phosphate, these are inhibiting hexokinase. Again, in case of phosphofructokinase, AMP, ADP, these are stimulating phosphofructokinase, whereas ATP, citrate and glucagon, they are inhibiting. Now coming to the pyruvate kinase. In this pyruvate kinase, what does this mean? It means that pyruvate kinase, when pyruvate kinase plus phosphate, it means pyruvate kinase, when it is phosphorylated, then it becomes inactive. Generally what happens when an enzyme is phosphorylated, it becomes active. But in this case, pyruvate kinase, when it gets phosphorylated, it becomes inactive. And pyruvate kinase, when it gets dephosphorylated, it becomes active. Now, glucagon is making the pyruvate kinase inactive. Yes, obviously it will make the pyruvate kinase inactive. What happens? Glycolysis is occurring, that means cell is utilizing the glucose. There is uptake of the glucose. But glucagon will not result in uptake of glucose. Glucagon will increase the blood glucose level. So this glycolytic pathway needs to be inactivated if glucagon is acting on it. And how can it be inactivated? By inactivating the pyruvate kinase, because pyruvate kinase is the final step where pyruvate is being produced. So glucagon is preventing the glycolytic pathway by inactivating the pyruvate kinase. And how is it inactivating the pyruvate kinase? By phosphorylating the pyruvate kinase. Similarly, on the other hand, insulin is promoting the glycolytic pathway. How is it promoting the glycolytic pathway? By activating the pyruvate kinase. And how is it doing so? By dephosphorylating the pyruvate kinase. It is activating it, and in turn it is activating the entire glycolytic pathway. I think the entire part is very clear to all of you, and glycolysis is now in your fingertips. Stay tuned. I'll be also coming up with different biochemical cycles, and stay tuned with this channel and share with your friends if you feel that it's worth sharing. Thank you.